Welcome back, ladies and gentlemen. So last time we talked about how eukaryotic cells, multicellular organisms to be more specific, are capable of adjusting their gene expression based on the condensing or relaxing of their DNA molecules. Today we're going to pick up with sort of what would be the next logical step in control, and that is the regulation of transcription. So remember, transcription is taking DNA to messenger RNA. So those chromatin modifying enzymes can provide initial control of the gene expression by making that region of DNA either more or less acceptable or accessible to the binding of your transcription machinery. So when we talk about that transcription machinery, we're really talking RNA polymerase plus a handful of other molecules that help that RNA polymerase to bind appropriately to the promoter region of a gene. So when we look at eukaryotic genes, right, we've talked in the past at this point about that promoter region, your RNA coding region, which would include your exons and introns, and then downstream of that would be that terminator region. If we go the opposite direction and we go upstream of that promoter, you also have these things called proximal control elements as well as enhancers, or you may also come across it as distal control elements. Those control elements are segments of DNA that don't ultimately code for any sort of protein information. They're non-coding sequences of DNA. Historically, we kind of thought this was just junk DNA, because it didn't code for a protein. Well, now we're starting to understand that no, in fact, a lot of that quote unquote junk DNA actually does have function, some of which are for these control elements. So that non-coding DNA helps to regulate transcription by being basically binding docks for other proteins that help to ensure that that RNA polymerase binds appropriately then to that promoter and enhancing its ability to actually read through the RNA coding region. So both those control elements and the proteins that they bind are really critical to the precise regulation of gene expression in certain cell types. So let's talk transcription factors, which can be some of those binding elements. To initiate that transcription, eukaryotic RNA polymerase requires the assistance of these proteins called transcription factors. These general transcription factors are essential for all protein coding genes. And really, it's a ability to just get everything in its proper place to fit on that promoter region of that DNA to ensure the efficiency of that RNA polymerase being able to open up the DNA double helix and actually effectively read through that RNA coding region and make that messenger RNA. So when we look in eukaryotic cells, that high level of transcription of certain genes really is dependent on these control elements interacting with these specific transcription factors in terms of regulating that gene expression. So again, we can have proximal control elements versus distal control elements, or what is also known as enhancers. So those proximal control elements are going to be found relatively close to the promoter region itself, so they'll be just upstream of that. If we go further upstream, that's where we can find those distal control elements or those enhancers. They can not only be far away from the gene, they can also be located in intron sequences as well, which is kind of unusual. But remember, those introns are interrupting sequences. They're not actually containing information for the protein production. So this is another place that we can also find those distal control elements. When we look at the way that this all works, so those enhancers, those distal control elements, will bind these proteins that are also produced by some other gene somewhere else. Those activators bind to those distal control elements. And what this allows this to do is basically hairpin loop our DNA onto this sort of transcription initiation complex. Now, the binding of those activators are caused by mediator proteins that interact with the promotings at the promoter region. So we get these sort of general transcription factors. We also get this DNA binding protein that comes into play here, your RNA polymerase, and then those activators. So in order to ensure that that RNA polymerase is bound properly to that promoter and allows this whole thing to work tightly, we're going to hairpin loop that DNA molecule, and those activator proteins then attach to this group of mediator proteins that basically cinches that RNA polymerase into place and makes sure it can't go floating away. Now, some of those transcription factors can also function as repressors. So here we're looking at activators, right? We're enhancing the ability for that RNA polymerase to bind to that molecule and increase RNA synthesis. We can also have the opposite, where some of those transcription factors act as repressors and inhibit the expression of a particular gene. Some activators and repressors can act indirectly by influencing chromatin structure as well, in terms of adding things like those methyl groups and things to the DNA or to those histones to help promote or silence transcription. So it ultimately is the combination of all of these things, these enhancers, the promoter, the distal control elements, that combined end up controlling the activation of transcription only when it's appropriate. So if I take a look here, remember we look at your cells. Every cell in your body contains all 46 chromosomes worth of information. So if I wanted to pull out a skin cell, I would still have the instructions to be a skin cell there. That would be what we would expect to be active. That being said, you still have the instructions to make cardiac muscle function, to make a neuron function. But in that skin cell, that set of information is silenced. So that's what we're looking at here. I've got the same set of genetic information, right? Here is an example of an albumin gene, which is something we typically associate with liver function, and then the crystalline gene, which would be typically associated with lens cells in your eyes. So in a liver cell, those active activators, those available activators, are attached to those enhancers. We're creating that initiation complex and allowing the transcription of that albumin gene to be expressed because we need that protein in those liver cells. If I look at the crystalline gene, however, I have a repressor bound to one of those distal control elements, and as a result, no hairpin loop, no binding of RNA polymerase, and so that crystalline gene is not being expressed. I go to a lens cell in your eye, however, and it's the reverse. So the repressor is bound to the albumin gene and not expressing that information because I don't need that protein in that cell. However, I do need the crystalline gene. So now I have activators attached to those distal control elements. I create that initiation complex, and now that crystalline gene is being expressed instead. So in eukaryotes, each gene has its own set of promoters and control elements. Those genes can be relatively close to those control elements, or those genes can be scattered across a bunch of different chromosomes. But each has the same combination of control elements. So I can use similar activators or similar repressors. I just need to attach them to the right promoter regions, the right control elements, in order to activate or inactivate the genes that I need in that particular cell. So copies of activators recognize specific control elements and promote the simultaneous transcription of those genes that are necessary collectively for that cell to be able to produce a set of proteins that it needs in order to do its function at that particular point in time.
Polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.